Now, in case your research entails reporting the amino acids interacting with the ligand, this is quite easy because you may actually want to compare the amino acids that were involved in the co-crystallized interaction and then the one after docking, which most times is scientifically valuable. So, since you already now know how to select this site sphere, simply come to the second option here, define and edit binding site. We can close this one so that it's not so boring. Then we can just click this. Oh, sorry, let's make it like this. Okay. Show and hide site sphere. Yeah. So now we can see that interactions between the ligand and the protein were tyrosine 9, arginine 13. So if you scroll through the entire amino acid sequence, those that were in or that are interacting within the defined distance are all highlighted up to phenylalanine 22. So you can note these amino acids and then use for your comparison or even use to set your interacting amino acids in the course of your work. We have also set our ligand now in Mo2 format. So we are making progress with our work. We also have saved or taken note of the binding coordinates, that's it, active site coordinates. So we can come back now to AutoDock, and we already have our receptor here. So let's set the grid parameters. Or first, let's bring in our ligand so that we don't have to go forth and back. Open. We already set the working directory. Of course, nothing will show because it's showing PDBQT. So click the drop down arrow, all files. Then you can see your Mo2 or select Mo2 alone, and then you are good to go. So we now have the ligand uploaded to the working interface, and then you have the Gestager charges already added. So if we check, you can see that yes, it is there. Then come back again to ligand, torsion. First, you want to detect the torsion root. We can bring that a little closer so that we can see that. Torsion talks about flexibility. So let's detect the root. Yeah, root detected. Then we come back into torsion, set number of torsions. So here you can try to set the number of rotatable bonds that you have within the molecule. But we'll leave it as it is now, standard. Dismiss. Like I said, you can actually increase if you wish or reduce. In case you want to do rigid ligand, rigid receptor docking, once you come to this set number of torsions, you take it to 0. But for this case, we'll leave it and allow it to stay flexible. Then finally, torsion tree, you wish to show set number, choose torsions, 5 of the possible 32, then done. So once you have done that, we will now go on to save the ligand because we also need it in PDBQT format. Output, save in PDBQT format. So the extension has changed, file extension PDBQT. Now it will be slightly charged, ready for docking. So we can come do the same now for the receptor, grid, macromolecule, let's choose 1GSF, select, okay. Also, we save it now as PDBQT. So we now have both charged molecules ready for the whole process. So in this first case, we'll go with the rigid receptor, and flexible ligand docking. So back to grid. Now choose. We already passed through this. Okay. Grid. Now set atom types. Choose ligand. Choose ligand. Select. Done. Back again to the grid. We have grid box. So this is where we now set our grid to actually cover the active site of interest. We are looking at site-specific docking, not blind docking. This is why it's important to understand how this grid works. So we have already saved this information somewhere before. This is our X, Control C. I'll just paste that here, Control V. Then next we have the Y. We copy this. And next we have the Z. You can see how they are moving, and then the box is changing position also. Here, Control C, Control V. So I left the so we can now also increase the dimension so that it can cover the area of interest. So after increasing the dimensions, right click here and set this to 0.5. 0.5. Apply. So normally we do this to ensure that there's more room for compounds to dock. Then you can see that it is now conveniently covering where our compound was initially docked. So you can now click OK. Then you come back here, File, Close Saving Current. So we have saved the grid specifically for this particular interaction. So come back again to grid, output, save grid parameter file. So it takes you back straight to your working directory. We can just use 1.gpf and save. So our grid is ready. But if we go back to our working directory, the AutoDock will not run if we do not have the AutoDock and AutoDock executable file and AutoGrid file. So if you go to your local directory, go to Program Files, go to the Script Research Institute, you will see here I have Vina, I have AutoDock because I've installed both. Then just click, copy these two files, AutoDock and AutoGrid, Control C, then bring them to your working directory. Just paste here. Then you can come back to AutoDock. You say run. Let's run AutoGrid now because our grid parameter file is ready. AutoGrid path name. Where will it find AutoGrid? This is AutoGrid here. Select open. Then next we have the grid parameter file. We have just this. Select. Automatically the file name is uploaded. So we're going to have an output of 1.glg read log file. Then leave other things as default and just launch. So once you see this, it means it is working. You wait a few minutes for it to run. Once completed, the dialog box closes. Then we are now ready to go. But before we continue, let's confirm what transpired. So we can see now we have several map files and other components Then we have the GLG file. So you must ensure that these ones, they are not empty. If you have 0 kilobytes, you will have issues with the docking later. So now that we have done the grid, sorted the grid issue out, next is to go to docking. Again, we're going to select our rigid file name. In this case, take note because we'll come into flexible residue file name later. Rigid file name, we're selecting our 1GSF-PDB-QT. Back again to docking, ligand, we're choosing, this time same itaclinic acid, select ligand, and then everything default, accept. Back again to docking, search parameters, we use genetic algorithm. I think we explained this in another video. Then number of GA runs. The more runs, the better the clustering. So we will take 50 and increase population size to 300. Because of PC capacity, we will leave the other ones like this. But with these two numbers, we should have increased this already. But we will leave every other thing as it is. Accept. Back again to docking. This time we have to now, the docking parameters, they're just there, just to show it to you. Accept using default. Come down to output, Lamarckian GA. Then we save this as a DPF file, docking parameter file, 1.dpf to make it easy. So click save. So once we have now saved the docking parameter file, we can go to our working directory again to confirm if we have a DPF. Yeah, that's it, 1 DPF, 3 kilobytes. So we can now come to run. This time we're running docking, run AutoDock. So we're going to select the program path name again. We know where we have it down here, AutoDock execute. We used AutoGrid the first time. Now we're using AutoDock. Open. Then our DPF file, one here, open. And then we're expecting a DLG, docking log file. So launch. Now, once you have this dialog box, means it is processing. You can confirm that by looking at your, what's it called now, your working directory to see if a DLG file has been, yes, 1.dlg is 0 kilobytes. So the docking has started already. And this may take quite some time because of the number of GA runs. If you select 10, it will work faster. If you select a higher number, it will take a longer time also to run. Just ensure that in your system you have power enough to continue the process till it is ready. Then you can actually open even the 0 to confirm if it is running. Open with Notepad, and then you can see that yes, it has started, setting up library and what have you. So it's already running. So we'll just come back when the process is completed. It can take several times depending on what you're working on. So once the docking process is completed, the other window automatically disappears. Now we can go to our working directory to confirm. So we have the DLG file. Now we open. And we can now go right to the bottom. Okay, successful completion. Yep, it took quite some time anyway. You have to be patient. 21 minutes plus.